Hello viewers, in my previous two lectures on DNA, I shared with you the chronology of events and discoveries that led to the elucidation of nucleic acid structure. The macromolecule, beginning from the work of Liebig, who coined the term inosinic acid, the separation of nuclein from the pest cells was done by Miescher. The macromolecule was finally named nucleic acid by Altman. It was Kossel who first found that the nitrogenous bases adenine, thymine, guanine and cytosine were components of nucleic acids. Later, he also discovered uracil. By this time, it was very natural to believe that nucleic acids were not very strong candidates to function as genetic material since they had such a simple structure as compared to proteins, which were far more complex with 20 amino acids. It was the pioneering work of Levine that revealed the presence of deoxyribose sugar in nucleic acids, thus leading to the naming of DNA. Levine also identified the ribose sugar and finally formulated the basic components of nucleic acids as nucleotides, which were found to be composed of a base, a sugar and a phosphate group. On your screens, now you can see the structure of a typical nucleotide where we find that a sugar molecule is attached to a phosphate at C5' and a nitrogenous base is attached to the C1' of the sugar molecule, and in this case the sugar molecule has been taken to be deoxyribose sugar, and this sugar can also be ribose sugar. However, Levine's tetranucleotide hypothesis for the structure of DNA was found to be erroneous and thus rejected. The interest in nucleic acids and DNA was rekindled when three scientists, Avery, MacLeod and McCarty, gave a convincing biochemical proof that DNA is the transforming principle and may function as the genetic material. Many scientists made notable and path-breaking contributions to the structure and nature of nucleic acids. Chargaff demonstrated the equivalence between purines and pyrimidines, that is, adenine is equal to thymine and guanine is equal to cytosine. In other words, the percentage of purines is equal to percentage of pyrimidines. However, you have to remember that the ratio of A plus T to that of G plus G, G plus C is constant for a given species, and this I have discussed in my previous lecture also, that species in which the percentage of G plus C is greater are more thermostable. For example, bacteria are more thermostable as compared to human beings. Why is it so? Because there is triple bond between G and Cs. Now, Chargaff's base pair rule was very important because it contributed a lot towards the elucidation of the structure of DNA. Four major groups of researchers conducted X-ray diffraction studies to find out the DNA structure. These were William T. Astbury, Linus Pauling, Wilkins and Rosalind Franklin, J.D. Watson, and F.H.C. Crick. The double helix model of DNA proposed by Watson and Crick was the most acceptable one. No doubt their work took important clues from the results of scientists like Chargaff, Levine, Astbury, Pauling, and Rosalind Franklin. Francis Crick, James Watson, and Maurice Wilkins shared the Nobel Prize in Physiology or Medicine in 1962. Undoubtedly, the double helix is considered to be the biggest discovery of the last century. Having understood the timeline of events leading to the double helix model, let us today discuss the structure of the individual components of the nucleotides before we study about the double helix model of Watson and Crick. As you know, nucleic acids such as DNA and RNA are made up of simple units or nucleotides. Thus, nucleic acids are polynucleotides. You have to remember, and I think you know also, that a nucleotide is made up of three components. A 5-carbon sugar, that is a pentose sugar, and now these sugars can be of two types. In RNA, the sugar is ribose. In DNA, the sugar is deoxyribose. The second component are the organic bases. Now the organic base will contain nitrogen, and since it is containing nitrogen, these organic bases are also known as nitrogenous bases. And you have to remember that a nitrogenous base is made up of carbon, hydrogen, and nitrogen. Nitrogenous bases are aromatic heterocycles. What do we mean by saying they are aromatic heterocycles? That is, they are cyclic compounds in which one of the ring atom is other than carbon, and in this case it is nitrogen. And these compounds are aromatic in nature. That is, they have a delocalized pi electron cloud in them, which gives them unusual stability. The third component of nucleotides is a phosphate group, which is present as phosphoric acid, that is H3PO4. From H3PO4 we take the phosphate group. This has the same structure in all nucleotides. The nucleotides are formed due to condensation reaction between these components. So what do we have to remember here now? That phosphoric acid reacts with the hydroxyl group on the sugar moiety, resulting in a ester bond, and at the same time a base reacts with a carbon 1 of the sugar moiety through glycosidic linkage. So all the three are connected, and during this process it is a condensation reaction between them, that is, small water molecules are eliminated during the course of reaction. Now let us discuss these three components in detail, that is sugars, bases, and phosphoric acid. Now, depending upon the sugar they contain, the nucleotides are of two types. That is, we can have a ribonucleotide. You have to remember, ribonucleotide is present in RNA, and this contains pentose sugar ribose, whereas in second ribonucleotide, the sugar present is 2-deoxy-2-ribose, which is again a pentose sugar. So what is the difference between a ribose sugar and a deoxyribose sugar? The difference lies between the groups present on the second carbon. What we find in the second carbon in ribose, we have a hydroxyl group, whereas in deoxyribose, the oxygen is removed and only hydrogen is present. Therefore, this sugar is known as deoxyribose sugar. The difference in the sugars affects the secondary structure and stability of DNA and RNA. Now, as you can see on your screens, to make it clear further, I've shown you the two structures of the ribose sugar. The first structure, which is on the left hand side, this structure represents the Fisher's projection formula. And what do we find here? That this ribose sugar is a 5-carbon sugar. It has 4 hydroxyl groups and an aldehyde group, and it has 3 chiral carbons in it. The difference will lie here when we compare it to deoxyribose. You will find that in deoxyribose, at this carbon we will have only hydrogen, oxygen will not be there. But this, and we call this structure as D. Now D does not refer that this molecule is dextrorotatory. This refers to the configuration of the OH on the highest numbered asymmetric carbon. And what do we find? That in open chain structure, the OH is on the right hand side. So this structure was given configuration capital D when it was compared to glyceraldehyde. But this structure could not explain all the properties of sugar, that is ribose, or we can say not all the properties of monosaccharides. So what do we find? That it undergoes intramolecular reaction, that is a OH on the 4th carbon reacts with a carbonyl group, the carbon of the carbonyl group resulting in a cyclic structure. And in this cyclic structure, what do we find? That this has a furanose ring. We call it as a furanose. Why do we call it as a furanose? Because the basic ring structure here is that of cyclic ether furane, and we call it as O's because this is a carbohydrate. And this is now the furanose form of O-ribose. And what do we find? That the orientation, that at the second carbon we have OH group, and we are calling it as the beta form, and beta refers that the OH is on the same side as CH2OH group, and D refers to that this molecule has configuration capital D. All this I have discussed in my previous lectures on configuration of monosaccharides in great detail. So you can go back and refer to those lectures also. Now what we find, how does this deoxyribose sugar differs from the ribose sugar? So here you remember please that D is referring to the configuration. What is this 2 referring to? It is referring that it is at the second carbon that we do not have a OH group. Oxygen has been removed from here, hence it is called as deoxyribose, and we have a hydrogen here. So this is the structure of beta D 2-deoxyribose furanose. And you know why are we calling it furanose? Because this structure is, basic structure is that of a cyclic ether, which is furane. Now let us, to summarize and to make more clear, I have taken another slide and I am sharing it with you, that when we are talking of this molecule and when we are talking it in reference to the nucleotides, you have to remember the numbering of these carbons will be done by primes. That is, we'll have 1 dash, 2 dash, 3 dash, 4 dash, and this is the fifth carbon here. And it is at the second carbon we'll find the difference between ribose and deoxyribose. In ribose, at the second carbon we have a OH group. So it is called as ribose. But in the second molecule what we find that the O has been removed from the OH group which is present at the second carbon. So we say 2-deoxyribose. So this is the difference between the two sugars, and ribose is present in RNA and deoxyribose is present in DNA. This little difference in configuration only at the one chiral carbon affects the properties and the stability of these two molecules. So what you have to remember that at remaining carbons the configuration is the same, the groups are the same. It is only at the second carbon where there is difference, that in ribose we have a hydroxyl group, whereas in deoxyribose we have a hydrogen. Now let us discuss the nitrogenous bases, which are the second components of the nucleotides. So what you have to remember here is that there are two types of nitrogenous bases. They can be pyrimidines and purines. These bases are nitrogen containing heterocycles whose rings contain both carbon and other elements, because we are saying it is a hetero. It is containing hetero elements. So in this case, the ring will contain carbons as well as atoms of nitrogen in the ring. So on your screens, now you can see the structures of purines and pyrimidines. Here I would like to first discuss the structure of pyrimidine. So before we go on to discuss the structure of pyrimidine, please remember when we were numbering the sugar moiety, we were numbering them with primes. Whereas when we are numbering the cyclic carbons, the carbon atoms of purines and pyrimidines, we are numbering them in simple numbers, that is 1, 2, 3. That is, they are not 1 dash, 2 dash, 3 dash. This is done to distinguish between the atoms which are present in the sugar molecule and the atoms which are present in the hetero molecules. Now when we look into these structures, when we look into pyrimidine, what we find that pyrimidine has 2 nitrogen atoms in it which are present in the ring. And when we start the numbering of this ring, which I shall be discussing in detail little later on, we will always start numbering from the lower nitrogen. That is, we will give it the number 1. And now we will follow the system in which it says that the lowest number to the first point of difference where this nitrogen will get the lowest number. So if I number this chain ring from this side, that is 1, 2, 3, 4 and 5. That is, if we number it from here, then what we find that this nitrogen is getting number 5. And if we number it in a clockwise direction, what we find we are getting here number 3. So between 5 and 3, which is lower number 3. So please remember, whether it is clockwise or anti-clockwise, you have to remember that is, we will follow the first point of difference. So these nitrogens should get the lowest possible number, and the nitrogen at the base will be always numbered as 1 in pyrimidine. And when we look at the structure of pyrimidine, what we find is that this structure is very similar to benzene. That in this case, if this nitrogen was CH and this nitrogen was again CH, that means this would have been the structure of benzene. That means this pyrimidine is quite stable because in it each atom is sp2 hybridized. That means it is having a delocalized pi electron cloud, and this is aromatic in nature. Now when we go on to purines, what we find, purine is made up of two rings. The bigger ring is the pyrimidine ring, whereas the smaller ring is the imidazole ring, and again we find that this structure is aromatic in nature, and we will start numbering from here, that this nitrogen will be given number 1, and then we proceed to number 2, 3, 4, 5 and 6, and then we go to 7. So what do you observe in these two structures? That always odd numbers are given to nitrogens. That is, nitrogens are present at odd numbers: 1, 3, 7, 9. Similarly, here nitrogens are present at 1 and 3 positions. I'll be discussing the naming and numbering in detail in quite shortly. Now let us see what are pyrimidines. I have explained the basic structure of pyrimidines and purines. This is the general structure. This is the structure which will be present in DNA and RNA of that purines and pyrimidines, but this will be slightly modified. That is, we will have some other substituents present on the ring. So let us see what are these and what do they result in. So what are pyrimidines? As I have already said, that pyrimidines are simple aromatic compounds composed of carbon and nitrogen atoms in a 6-membered ring, and we've already discussed the molecular formula of this ring will be C4H4N2, and that means the structure is very similar to that of benzene, because we know that the molecular formula of benzene is C6H6. So what have we taken here? That is, instead of taking 6 carbons here, we are taking 4 carbons, and 2 of the carbons of the ring have been replaced by nitrogen atom. So pyrimidine, after this discussion, we know is a simple aromatic ring composed of 2 nitrogen atoms and 4 carbon atoms with hydrogen atoms attached to each carbon. So this we've already seen. The carbon and nitrogen atoms are connected via alternating double and single bonds, and this bond structure allows for the resonance or aromaticity, causing the ring to be very, very stable. So in my next lecture, now I shall be discussing the derivatives of pyrimidines, and then we will go on to discuss the purines and their derivatives. For this lecture, I had consulted the books by Biochemist by Lehinger and Morrison and Boyd, and also books by Brucker Robert, that is Concept of Genetics, Nelson David, Cox and Lehinger, as I've already said, Benjamin's Genetics, a Conceptual Approach, Klug, Williams and Cummings. Thank you.